Okay, it's just hit 2 minutes past 4 CAT on my watch. I understand that it's 9 a.m. EST and different times, depending on where you are in the world. Good morning, good evening, good afternoon, depending on where you are, as I say, because we're an international community, so I understand that we have audiences from across the world. Thank you so much for joining us for the February webinar. So this is the second webinar for our webinar series for the IHCC. And we host monthly webinars. So there's a team of us in the background, including Ludi, Latifa, Farah, and other people that are organizing these webinars. And I'm really honored today to have two colleagues based in Africa and elsewhere in the world that are going to be presenting on, as the title of the talk says on the slides, is Human Diversity in Africa, or the Novel Coronavirus Host Susceptibility Study in South Africa. So we have two speakers today. One of them I know very well. She's a professor actually, but I'll say doctor here because that is what the website said, but she's a professor. Professor Michelle Ramsey, as well as Dr. Andrew May. So I'm just going to be rid of their bios. If you want to read the abstract, you might have to find the ad that we sent out on Twitter, on our website, and elsewhere as well. But I'm looking at time and I don't want to take too much time. But Michelle has done so much, so I might take a bit more time reading her bio as well. Dr. Michelle Ramsey is the director of the Vet Sidney Brenner Institute Molecular Bioscience, and she's a professor in human genetics and the South African Research Chair in Genomics and Bioinformatics of African Populations. Her research aims to shed light on the role of African population genomic variation in susceptibility to diseases in the context of the ethnic as well as environmental diversity across the African continent. She's the PI of the African longitudinal cohort of over 12,000 individuals as part of the H2Africa Consortium. And she's also on the Scientific Advisory Committee of the H2Africa Bioinformatics Network, HJ-Bionet. She's also an external advisory board member of the EU-Africa PERMED Consortium, and she's a member of the International 100,000 Cohorts, IHCC. Professor Michelle is the president of the International Federation of Human Genetics Societies. Her recent awards include DSI Distinguished Woman Researcher, which she got in August 2019, NSTF South Africa or South 32 Lifetime Achievement Award, which she received in July 2020, as well as the gold medal from the South African Medical Research Council, which she received in March 2021. And there's much more, so please Google her when you have time if you want to learn more about what Michelle has done in her career. The second speaker, who's equally great, is Dr. Andrew May, who's an early-stage postdoctoral researcher situated at Sidney Brenner Institute for Molecular Bioscience. And he also works with Professor Michelle, hence they're going to be presenting together today. He's also affiliated with the Neuropsychology Research Laboratory at the University of the Witwatersrand in Johannesburg. He holds a Master of Science in Medicine and Human Genetics, as well as a PhD in Research Psychology. His research interests focus on the intersection between genetics and psychology, especially in physical and mental health, as well as childhood development. I'm not going to waste more time introducing our speakers. I just want to say thank you so much to both Michelle and Andrew for taking time out to give us a presentation to the IHCC community. We're really looking forward to it and we're excited. Over to you, Michelle and Andrew.
So thank you very much, Pabalu. I'll start us off and just to say welcome to everybody to this webinar. It's always a pleasure to be able to tell you a little bit about the work that we do. So when we were first invited to talk about the IHCC pilot project, Coronavirus Host Genomics Study, the COVIGen-SA, I thought that it was important that I talk about the link between the cohorts that we are building and this particular pilot study. So first of all, I want to tell you a little bit about AWI-Gen, which is really the cohort that is registered under IHCC. And what you will see is that that COVIGen-SA stands apart from it, that they're not actually linked at the moment, although it is our intention to link them. And what binds them together is the fact that they both run out of the Sidney Brenner Institute for Molecular Bioscience at the Faculty of Health Sciences at Wits University in Johannesburg. So both of these cohorts are part of the IHCC. And of course, the IHCC is huge and multi-national and global. And these are just two of the small cohorts linked to it. But the other connection that we have within the IHCC is that both COVIGen-SA and AWI-Gen are part of the SAPRIN network. And what we're really hoping to do is to link these different cohorts together and to use the opportunity that we have through the IHCC to do this and to thereby amplify both these projects. So that's really our objective. So I've just got a few slides on AWI-Gen because I thought it would be good to give you a little bit of context, but just to say that we do in the newsletter also have a profile, a cohort profile coming up, and that gives you links to some of our papers and more information. So first of all, where does this odd name comes from, AWI-Gen? So it's really an acronym where it's for the Africa WITS, which is our institution, in-depth partnership, where in-depth is an international network for demographic health and surveillance centers across low middle income countries. But AWI-Gen itself is a cross-sectional study, and its aim under the banner of H3 Africa is really to understand the genetic and environmental contributions to cardiometabolic diseases and related traits in African populations living on the continent. And as you can see, we work in 4 different African countries. You'll see that we started this project in August 2012, which was really the start of the H3 Africa consortium. And initially were funded for 5 years, but are delighted that we've got funding for another 5 years to actually expand this cohort, which we think has enormous value despite its rather modest size in the context of the IHCC consortium. So the next slide shows you the map of where we have 6 different research sites in 4 different African countries. And our baseline data collection occurred between 2013 and 2016. And what you can see here are the participants between the ages of 40 and 60. And as I said, it is a population cross-sectional cohort, so we didn't select participants based on having a particular disease or being a control who is disease-free. It was really using a random sampling frame to look at that age bracket. And of course, we chose it because it is relevant to cardiometabolic disease. That is what we are studying. So the entire cohort is over 12,000 individuals. And you will see that in Agincourt, we have several individuals over the age of 60 because we harmonized with the study that they were already conducting. So in AWI-Gen phase 2, we are converting this into a longitudinal cohort where we will have a second wave of data collection between 2017 and 2022. And we've added several novel components because I think, whenever you go in for a renewal of a grant, you want to make sure that it's fresh and that you are going to be doing something new. So we embedded in AWI-Gen a microbiome study, which is being done. It's the gut microbiome across a subset of individuals, but in each of our 6 study centers. And that is being led by our colleague, Scott Hazelhurst. And they've already had 2 publications from the microbiome work. But if there are people on the call who are interested, we're always looking for collaboration and partnerships. Then we've also decided to look a lot more at interactions with infectious diseases, but also to look at the menopause transition amongst the women of AWI-Gen, where we're looking at hormone levels and also at epigenetic changes that may have occurred across this 5 or 6 year span between wave 1 and wave 2. And we've also now focused on collecting data related to cognition, but also frailty and other elements. So very interestingly, the cognition data is now becoming very important also in the context of IHCC, where there is the Davos Alzheimer's study, where we could start looking at some issues related to cognition and dementia. So that's given us an opportunity for using this cohort for different purposes to which we collected it in the first place. So the next slide shows you where we were in December of last year in terms of recall of our study. So this is something which is always very tricky in longitudinal studies, that trying to find all the participants to study them again is always very complicated. And we had set a goal for ourselves to have a recall of about 70% of the cohort. And you can see there that some of the centers, the first 3 who collected their second wave of data and completed that process, Soweto managed to go back to 72% and the others just under the 60%. So reaching that 70% target is indeed very difficult. And in this age group, we have attrition due to deaths in the cohort, but also due to out-migrations or to unwillingness to participate in a second wave of collection. And, sometimes people are hesitant about giving more blood samples, or they don't understand if they're healthy, why they should be taking part in this particular study. So we are interrogating this loss to follow up. And we're hoping that the other 3 centers that are still in the field will manage to reach our 70% target. So the last slide is just, oh no, it's not actually the last slide, it's the penultimate slide. And this is to just talk about the richness of the data that we have on this cohort. So although our focus is on the cardiometabolic disease endpoints that you see there, we've collected an enormous amount of data. And specifically also to try and understand mediators and potential confounders in trying to link, for instance, genetics to cardiometabolic disease. So we've collected data on demography, including education, socioeconomic status, and also on behavior and exposures, because we know that these are very important in terms of cardiometabolic disease. And then we've looked at infection, because again, we feel that one shouldn't look at just one aspect of a person's health, that it is influenced by so many different things. And then we've done a lot of measurements, and we've collected blood and urine and done biomarkers. But I think what is rather rare in our population is that we used ultrasound to look at visceral and subcutaneous fat, and also at the carotid intermediate thickness. And these, of course, are markers of some forms of cardiovascular disease or risk factors for cardiometabolic disease. So I just want to talk briefly about the genetic data that we have attached to our cohort. So we have genome-wide genotyping data on just over 10,000 individuals, and we will be doing it for the full cohort soon. And that was done using the H3 Africa array, which is enriched for common African genetic variation. And this is also what we've used in the COVIGen study, which you'll hear about in a moment. And then we have whole genome sequencing on 160 individuals. And this, of course, has allowed us to do a lot of population genetic studies, some of which have been published. And currently we're having whole genome sequencing done on another 750 individuals as part of another partnership that we have with a commercial company. But we will also plan to do regional sequencing of areas of the genome of particular interest. And I think this again will give us very interesting data. So I think the next slide, which is the last one, is just to show you that this is a very complex partnership. And of course, each of our in-depth centers has funding from many different organizations not shown here. But we hope that this is a cohort that will add value to the IHCC and will also inspire you to collaborate with us. So I think without further ado, I want to hand you over to Andrew May, who is a postdoctoral fellow at the Sydney Brenner and has been involved in the coronavirus host genomics study from its outset. And he will introduce you to the partners and also the members of the core team, some of whom are on this call. So Andrew, over to you.
Thank you so much, Michelle. Hello, everyone. A pleasure to be with you all today. We are having quite a thunderstorm here, so please excuse any background noise you might hear. But as Michelle mentioned, I'm going to be telling you a little bit about the coronavirus host genomics study South Africa, which we call COVIGen-SA for short. So as you well know, COVID-19 is now ranked amongst the deadliest global pandemics, and it has severely disrupted the status quo of our daily lives. And at the outset of the pandemic, continents that have the poorest healthcare systems infrastructure were anticipated to bear the heaviest disease burden. And there were, of course, particular concerns about Africa, with some forecasts suggesting that by June 2020, the continent would be the source of 70 million infections, along with over 3 million deaths. A little over 2 years later, however, available statistics suggest that Africa, conversely, has the lowest total case and death counts compared to other continents, in spite of the limited healthcare infrastructure in Africa. And this paradox has puzzled many public healthcare experts, and it's been the topic of much debate, both in the scientific literature and more broadly. Of course, one of the most often cited reasons for this paradox is the low testing rate on the African continent, with tests per 1,000 individuals in African countries being just a fraction of those that we see in developed countries such as the USA and the UK, for example. However, many scientists are not convinced that the limited testing rate in Africa sufficiently accounts for Africa's low COVID-19 disease burden. And this has led to many other explanations and hypotheses that have been put forward to try and explain this low burden. So in terms of demographic and geographic factors, we know that the COVID-19 disease burden is worst amongst the elderly, but Africa has the world's youngest demographic, with a median age of just 19.4 years. And this sits in stark contrast to Europe, for example, where the median age is around about 40 years old. Secondly, due to its central location close to the equator, we know that Africa enjoys a warmer year-long climate, and this creates rather unfavorable conditions for viral spread. And then thirdly, we know that many parts of Africa are characterized by a lower urban density, and this has been thought to limit the spread of the SARS-CoV-2 virus. However, this idea is not without contention, as there are some reasonable arguments to suggest that viral spread in rural areas can be equally problematic. For example, we know that rural households typically have more members than urban households. Self-isolation in a rural setting simply isn't as practical as it is in an urban setting. And because of the widespread job loss that the pandemic has caused, we suspect that many urban individuals are returning to rural communities, and this is helping to facilitate the spread of the virus. Then in terms of logistic factors, we know that Africa had slightly more advanced warning of the pandemic, and this allowed African countries to model their preventative responses and measures based on the experiences of other countries, especially in terms of lockdown measures. Moreover, because communicable diseases remain a substantial issue on the African continent, African countries have a wealth of experience and very well-established infection control measures, both of which have likely been leveraged to help combat the spread of COVID-19. And then lastly, in terms of possible biological explanations, due to the widespread use of certain vaccines, such as the BCG vaccine, continental Africans may have slightly better trained immunity compared to other parts of the globe. And then along similar lines, we know that Africa harbors the widest variety of bacterial, parasitic, and viral pathogens, including other coronaviruses, which creates quite a potent selective pressure. And this idea was most recently supported by a Nature publication in 2020, which included scientists from WITS and the Sydney Brenner Institute, in which signatures of selection in African genomes were predominantly detected for immune-related variants, including those associated with HIV and influenza. So while on the topic of genetics, it's thanks to the work of large consortia, such as the COVID-19 Host Genetics Initiative, that we're beginning to appreciate the substantial role that Host Genetics has in understanding the susceptibility and severity of COVID-19. In their most recent worldwide meta-analysis, the Host Genetics Initiative found 15 lead association signals throughout the genome, and some of these were as evident in hospitalized cases as they were in individuals with a reported infection regardless of symptom severity. Now, unfortunately, in this worldwide meta-analysis, African ancestry individuals only accounted for about 5% of the effective sample size, and none of these individuals were actually sampled from the African continent. Now, this is an unfortunate oversight because we know that genetic studies on continental Africans can be hugely informative, given their unparalleled rich genetic diversity, which affords a linkage disequilibrium structure that is characterized by much shorter haplotype blocks. And it's this advantage that was actually recently leveraged by Jennifer Huffman and colleagues in a paper published last month, where they particularly looked at this association signal on chromosome 12. So in all other ethnicities, this lead association signal co-segregates with a wealth of other variants, and this makes the identification of causal variants quite complex for this locus. However, Huffman and colleagues noticed that the same signal has far reduced linkage disequilibrium with surrounding variants in African ancestry individuals, and this helped them to confirm that this variant itself, this lead association signal, is actually a causal variant. It's a splice site variant, and the G allele of this variant helps to protect individuals from COVID-19 related hospitalization. And it just so happens that the frequency of this protective G allele is substantially more in African ancestry individuals compared to other ethnicities. So it's these sorts of results that suggest to us that genetic risk profiling for COVID-19 in African ancestry individuals is likely going to be quite different to other ethnicities. But unfortunately, all of our knowledge to date stems from non-continental African individuals who are likely to have a more admixed genetic architecture, and who also have a comorbidity disease profile that's different to continental Africans. And so we consider a host genetic study here in a continental African sample to have quite diverse benefits. So it's motivated by these sorts of reasons that the aim of the COVIGen-SA study is to provide a platform for COVID-19 research that accounts for individual variability in the genetics and health backgrounds of Black South Africans. And our two main objectives are to firstly conduct a multi-approach investigation into host genetic factors affecting COVID-19 susceptibility and severity. And then secondly, to establish a research resource of harmonized clinical and genetic data for a large sample of SARS-CoV-2 positive Black South Africans. In order to maximize our participant recruitment and the efficient usage of resources, we've partnered with a number of different clinicians and researchers who are all involved in a range of different studies that are either directly or indirectly related to COVID-19. And to date, we've partnered with 7 other institutes who are involved in 15 relevant studies. Our network of partners assists us with participant recruitment, which involves the donation of a blood sample from which DNA is extracted, as well as the completion of a study survey which captures pertinent clinical and demographic variables. Our DNA samples are stored in an approved biobank here at the Sydney Brenner Institute, and our study data is stored on a curated REDCap database. And together these form our COVIGen-SA research resource, for which we have a number of planned host genetic studies, including exploratory genome-wide analyses and targeted whole exome and whole genome sequencing. And these analyses we plan to do after stratifying our participants based on disease severity. The entire project is underpinned by a strong governance framework which promotes cross-disciplinary collaborations, transparent and complete ethical adherence, data and sample sharing, and public data accessibility. So just to speak to the methods in slightly more detail, our initial goal is to recruit 5,000 Black South African participants with a PCR-confirmed SARS-CoV-2 infection. We're currently recruiting participants from 10 different study sites that cover 5 of the 9 provinces here in South Africa. As Michelle mentioned, these case individuals will be typed on the Illumina H3 Africa array which consists of a panel of 2.3 million SNPs enriched for those variants common in African individuals. And we'll compare these case individuals against a population matched control that consists of the 5,000 Black South African members of the AWI-Gen cohort that Michelle described earlier, because they were typed on the same array. So just to speak to some very preliminary results from only a small selection of our partners, we have quite an eclectic sample to date. Female individuals currently hold the majority in our sample, as do young individuals below the age of 40. We are seeing that in our rural samples, the number of household members is much higher than in our urban samples. Interestingly, we have struggled somewhat to identify severely affected Black South African individuals with COVID-19, and this possibly recapitulates our earlier discussion about the low COVID-19 disease burden here in Africa. And then of the many different comorbidities we've captured on our cohort, I've just highlighted here the number of HIV positive participants, because this is another factor that potentially distinguishes our sample from other global samples. And it's also an avenue for further research, because there has been conflicting evidence about the interplay between HIV and COVID-19. We have genotyped a single batch of our participants thus far, and through early principal component analysis, we see the typical loose clustering that characterizes African samples compared to other ethnicities such as European, Indian, and Asian. When we look at just our case and control individuals thus far, we do see a decent overlap. But again, that very loose clustering both speaks to the rich diversity of continental Africans in terms of their genetic makeup, but it also speaks to some of the difficulties of doing genetic studies in these types of samples, because one has to be much more cautious in the sorts of quality control thresholds and the analyses that can be conducted in order to account for this wide genetic variability. So just to summarize some of the achievements of the COVIGen study to date, as I mentioned, we have 7 different institutional partners involved in 15 different studies. Ours is quite a cross-disciplinary effort, including clinicians, geneticists, epidemiologists, and a bioethicist. We have 10 recruitment sites across 5 provinces. We have over 2,000 participants enrolled to date. We have 1,152 participants with genome-wide genotypes, and we hope to begin our first genome-wide association study in April this year. And we also have a cohort profile paper that describes our COVIGen sample in a little bit more detail that is nearing submission. So just to end off, I'd like to acknowledge the other core members of the COVIGen-SA team, including Dr. June Fabian, Dr. Harriet Etheridge, and Heather Seymour, as well as all of our clinical and research partners. In terms of funding, we are tremendously grateful to the IHCC, whose funding helped to kickstart the COVIGen-SA project, which has now gone on to grow from strength to strength, thanks to the funding contributions of Alive and Illumina, as well as the funders of all of our different partners' studies. And then I'd also just like to thank the Center for Proteomic and Genomic Research, which is our service provider for the sample genotyping. And then lastly, I just want to thank all of you for your attention. Thank you so much.
Okay, thank you so much. Michelle, I don't know if you have another presentation. Take over for Q&A.
No more presentation, but happy to take questions. Thank you.
Okay, sure. Thank you so much. Sorry, my camera is a bit hazy. I tried to wipe it off, but it didn't work. But thank you so much for really great presentations, starting with Michelle. Andrew, really great study. I mean, I'm excited just listening to you. Can't wait to read the paper that's about to come. Already there's a comment and question in the chat. One is from Jeff. And Michelle, I think this is for you and Andrew. So Jeff Ginsberg says, would like to hear more about collecting environmental data, what data and how to link the participants. Thank you, Michelle and Andrew. That was from Jeff. In response, Michelle and Andrew?
Could I just add to that? Because I think potentially the combination of environmental data and genetic data might give a much more clear picture of infection and susceptibility, et cetera. So it's related to what you presented, but I was thinking about gene by environment interactions and response to the infection. That's where I was going.
No, that's great. Thank you, Jeff. So, you know, we often interpret environment in its sort of broadest sense. So the environment can be things like smoking. And, you know, when we look at tobacco exposure, we look not only at smoking, but also at chewing tobacco and snuff, because in Africa, you know, there are lots of cultural reasons why people do one or the other, but there's also a very interesting gender difference in terms of smoking, for example, in West Africa. But what we've done in the second wave is we've also asked a lot of information about household smoke exposure in terms of whether people cook inside or outside. So we're trying to get more data. We then are working with the group on pollution and air quality in 2 of our 6 sites. I think that's going to be really interesting. And then, you know, we've looked at pesticide exposure because some of the areas that are rural are the farming practices means that people are often very exposed. So we thought that that was important. And then we are looking at the infections, and hopefully we can add COVID now to the AWI-Gen as well. And, you know, the other thing that we can do is look at things like NASA and temperature, you know, across different regions. I think there's a lot that one can do with data that you haven't specifically collected, but that you can get from outside sources. So we're really interested in doing that. And as part of the DSI Africa consortium, we're hoping to look at more of that kind of data as well.
So thank you. One quick clarifying question. In your cohort, you can link geospatial location of the participants to the you know, to the environmental data. I assume you can, but I just want to understand how you make that linkage.
So Jeff, that is interesting because although we didn't collect that data, some of the in-depth partners who are HDSS sites have collected some of that data. So I think one of the challenges we have is to do data linking. So at the moment, you know, and for AWI-Gen, we've just asked them, you know, which cohort they're from, and we know that they're geographically quite close, but the specific location, we would have to do data linkage. And then of course, the issues of, you know, whether this is personally identifying information comes in as well. And being mindful of this new Protection of Personal Information Act in South Africa, we will have to do that in an ethically sound and legally compliant way.
Okay, thank you so much for those questions, Jeff. And Michelle, thank you for your answers as well. There's a comment from June Fabian. Thanks, Andrew and Michelle, great presentation. I have with that. Ludi has a very interesting question, and I think this is based on what Andrew spoke about. Would antiviral drugs given against HIV or other infections have also played a role in reducing the spread of COVID in Africa? Andrew?
So it's a really great question, and I can't speak to any specific research that I know about HIV drugs per se. I did just briefly see that there have been 2 studies here in Africa looking at the interplay between HIV and COVID-19, and the one study found that COVID-19 severity seems to be worse in HIV positive participants, but a study in Nigeria actually found the opposite and seemed to suggest that individuals who are HIV positive were somewhat protected against COVID-19. Now, whether this is due to the virus itself or because of the medication they were on, I'm not entirely sure. I would need to read that in more detail, but I think it's such an important avenue of research just because the HIV burden on the African continent is a serious concern. So it's important information that we need to uncover.
Okay, thank you so much for that, Andrew. We have about 2 hands that I see in the audience. Rongling, go ahead and ask your question.
Yes, thank you so much. Thank you, Michelle and Andrew, for this excellent presentation. And also, in terms of the paper that Andrew mentioned at your presentation about the Jennifer Hoffman's paper recently published, I just presented that paper at our division journal club. I'm so glad they are going to replicate this study and then replicate it to see if there's OAS1 gene that SNP is a causal SNP. And at the paper, I didn't see they mentioned any weakness or limitations, but you pointed out excellently. You look at the pre-existing clinical condition and look at all this the confoundings, the covariates, to look at it, to test if this SNP really causal SNP. So I think the IHCC has much more potential to do the science there, to replicate other study, and also to discover the new no matter genetic or environmental factors that affect the people's health, especially for this COVID-19. It's a hot topic for now. So I'm so glad you presented that, and that you are going to replicate that study. It's very good, very informative presentation. Thank you both so much.
Thank you so much for your comments, and I completely agree with you. I think the Huffman article was particularly informative, but again, it was limited by the fact that, as far as I could tell, all their participants were not continental Africans. They were from the USA and the UK. So as you say, it would be excellent to replicate that finding in a continental African sample and see what other novel variation we might find.
Okay, thank you so much for that. Thank you for your question and comment, Rongling. We have another hand. Karen, go ahead and ask your question.
Yes, thank you so much. Good day to everyone. I just had a quick question, and thank you for such an informative presentation. I know that you all are conducting targeted exome sequencing GWAS studies, and I was just curious to know, I mean, given the paucity of data among communities of color, for example, African Americans or Africans, not necessarily being represented in large-scale population cohort studies, with the targeted exome sequencing and whole genome sequencing that is being conducted, are you all targeting any known candidate genes, maybe distinct in certain subpopulations? For example, I know that their transcription factor 7, like 2, is a common gene that is mostly known across various ethnicities, but it's understudied in distinct African, or I should say populations of African ancestry. So I was just really curious to know if there were any targeted candidate genes that you all may have been interested in.
Yes, a great question, Karen, and I agree. I think there are some target regions that have already been picked out in the literature that would be interesting to explore in more detail. So as you mentioned, that toll-like receptor gene, that would be interesting to look at that area, that locus, as well as the area, the region on chromosome 3 as well, where that Neanderthal haplotype was discovered to be the leading risk factor amongst individuals of European ancestry. It would be interesting to see what happens there in African ancestry individuals. And I think also in terms of the role that blood group plays on the isoagglutinin locus, and whether that has any difference in African ancestry individuals. So we haven't formalized or narrowed down an exact targeted panel of candidate genes, but I think there are some definite candidates that we would like to look at. I don't know if Michelle wants to add some more.
Thank you, Andrew. Just to say that we are also doing capacity development in this project, and that we have a PhD student, Heather Seymour, who's also in the audience here, who will be, as part of her PhD, looking at some of these candidate regions. So we're hoping to get more information, and, just making the point, when you're looking at African populations, it's so important not to only use genotyping arrays, but to do sequencing, because there's so much variation that's not represented in an array, and your opportunity for novel discovery is really high. So we certainly will be doing that. Thank you.
Okay, thank you so much for your question, Karen, and Andrew and Michelle for answering that. I'm going to go to your comments quickly just to read one of the questions that I see from Ricardo. Great presentation. Looking forward to the results of the study. So the question is, Are you storing samples that may allow future functional genomics, for example, P, B, and C of buffy coats and RNA later, or proteomic studies? So that's for both Michelle and Andrew. Does anyone want to tackle that question?
So Ricardo, I'm not quite sure if you're referring to AWI-Gen or COVIGen-SA, but maybe I can answer it in two parts. Just to say, for AWI-Gen, we are storing samples. You know, we've got plasma, serum, urine, and then DNA. But unfortunately, we didn't collect samples for RNA, which is a limitation. But what is interesting, and Andrew may want to elaborate, is that our COVIGen-SA study is built on a lot of other studies, and some of the other studies or the other parts of the studies have collected samples and will be looking at many other aspects. So this is a great opportunity again for partnership, although our part of it is the host genomics. There's a lot of other research going on.
Yes, I'll just confirm what Michelle was saying. So far we are focused mostly on collecting the buffy coats and just doing DNA extraction, but as Michelle mentioned, our partners might have other samples to enrich the resource with.
Okay, thank you so much for that. I see there's another hand in the audience. Crystal, go ahead and ask your question.
Hi everyone, thank you so much for a nice presentation. My question is for Andrew. So how are you defining your COVID-19 severity cases versus the controls?
So it's a really good question because I know this is a point of contention in the literature. So in terms of defining our cases, we're trying to follow the guidelines set by the Host Genetics Initiative and how they describe severe, mild to moderate, and asymptomatic. So severe being those that were hospitalized in ICU or needed mechanical ventilation, and then mild to moderate is just presenting with symptoms. But then, of course, we're using a population control, which is a limitation because we can't confirm whether or not any of those individuals had COVID or not. And I know there is potential avenues, because they come from the AWI-Gen cohort, to perhaps return to them and try and get these data and see if they have actually been exposed to SARS-CoV-2. But it is a serious limitation because we won't know that, which will reduce the ultimate power of our association analysis. But obviously it is the strategy followed by a lot of other association studies worldwide, that you just, because you need such a big sample size to be well powered, you have to use a population control, and you do run that risk of not knowing exactly who in your population control is unaffected. I hope that answered your question.
Yes, thank you so much.
Okay,
it's quite interesting.
So
if I can I just
go ahead,
elaborate, Pabalo? So great answer. But you know, we kind of interested in you know, if we for the AWI-Gen study start phoning all the participants to find out about their exposure to COVID. In the West African countries where we work, in Burkina Faso and Ghana, our partners there tell us that they see very little COVID. So you know, we don't know whether it's a lack of exposure, whether it is because they're in such a rural area. So you know, I think it will be very interesting. But Crystal, you know, again, if somebody reports not having had COVID, you don't know whether it's an issue of exposure. But of course, the different waves also make a difference, because we know that the different you know, sort of COVIGen variants have different infectivity. So it is quite a fascinating thing to look at. And we're almost thinking we have to look at, you know, when they got the COVID. So I think there's so much heterogeneity and so much that we need to look at.
Okay, thank you so much for that answer, Michelle. Sorry for jumping in your mouth earlier. There's a comment from June Fabian saying there was a Spanish study that showed that TDF/FTC regimens are protective against COVID-19 infection as well as severity. We don't have the South African data regarding these, but first-line regimens in South Africa do contain TDF and FTC. I don't know if there's any response to that comment from Michelle or Andrew. The comment is from June.
So just to say, yeah.
Sorry. Go ahead, Michelle.
So June is the co-PI on COVIGen-SA. Maybe she can put on her camera and just say hello.
June, go ahead. June.
You're muted.
Always someone who's got to do it. Hi, just chipping in from the side. But yeah, we are hoping to try and unravel some of the HIV stuff. There was a population-based study, as Michelle mentioned, in Agincourt, which is one of the study sites, which is a rural study site. And they showed that just having HIV infection confers an odds ratio about 1.5 to 1.3 in terms of contracting COVID. But I think if you ask most clinicians on the ground, the sense is that it's much more people with comorbidities like diabetes and hypertension and cardiovascular disease who seem to be affected, as opposed to people who have HIV and are stable on treatment. So we'll see.
Any addition? Thank you so much, June, for that. Andrew, Michelle, any comments? Oh, there's a hand. I see a hand. Who's that?
No, it's my hand.
Oh,
The reason I put my hand up is I know that there are other colleagues in South Africa, and perhaps the Stellenbosch group who's on the call could tell us a little bit about the host genomic work that they're doing on COVIGen as well. You know, we often cast our net in our networks, but not beyond. And so hearing from other people, both South Africa, but other IHCC members who are doing host genomic studies for COVID would be really great.
So there's an invitation. Please go ahead, either raise your hand or tap in the chat box. Prefer if you can raise your hand and speak. We still have about 12 minutes left before we finish the webinar. So still engage. And I do have some questions of my own, but I'm giving the audience some time. Crystal, I see your hand is up. Go ahead, Crystal.
Hi, so just lowering my hand again. So I'm part of the Stellenbosch group, looking at COVID host genetics as well. Specifically, I'm focusing on a cohort in children that have an extreme phenotype called MIS-C. So we're looking at any human genetic variants that we can find maybe in these patients.
And Crystal, in terms of South Africa and our interesting demography, are you focusing on any specific group or all children with severe COVID?
So whoever comes into Tygerberg Hospital, we usually recruit. We're not excluding based on ethnicity, which is a bit of a downfall, but because it's such a rare phenotype we see, we can't really exclude children based on their ethnicity. Otherwise, the group will just be way too small. But most of them are of mixed ancestry.
Thank you.
Okay, thank you so much for that, Crystal. Any more comments? Oh, I see someone with an iPhone. Go ahead.
Yes, sorry for that. I am Dr. Nahla Afifi from Qatar Biobank. We have a cohort from our population already we did from the 14K whole genome sequence data. We identified that we have more than 2,000 participants already. They got COVID. So we have their whole genome sequence data is available. And also we have a cohort from when we recruited COVID patients around 3,000, but and we send the samples for the sequence, but till now, hoping that within few months they will be sequenced as a whole genome sequence. Those will be multinational, either they are Arab or non-Arab. They're from 36 countries. This is the COVID biorepository, which is available data. We have phenotypic data for them. We have a questionnaire data about the health. This data is available, but the genomic data still, we are waiting for the results, hoping within few months.
Okay.
That sounds amazing. Yeah.
Thank you so much for that comment, Doctor. There's a comment from Jeff. There are a number of investigators here from all of us or /US. Would encourage a replication study in African Americans if possible at some point in the future. Also replication studies in other cohorts via IHCC. I don't know if there's a comment to that comment. And there's a question from June asking Crystal about, is she also recruiting, or are they also recruiting from the Red Cross Hospital here in Cape Town? So if you can engage those two sort of comments and questions.
So yes, it will be encouraging for replication of the data for sure. And also we are willing for collaboration with different collaboration to access the data, because we can share data, but we cannot share samples. So it can be.
Okay, thank you so much. Crystal, go ahead.
Yes, so we do have, with regards to the question, are we working with Red Cross as well? So yes, we have a collaboration with UCT, and we are working with Dr. Kate Webb and Dr. D Abrams, sorry. I couldn't remember her name now. Dr. D Abrams from Tygerberg Hospital as well.
Okay. Thank you so much for that. We still have about 5 minutes or so left. I'll ask my questions if there's no more questions, but please, we still have time. So if you have a question, raise your hand or type in the chat box. So I have two quick questions. One is for Michelle and one is for Andrew, and I hope I remember them clearly. So the first one is about AWI-Gen, Michelle. You spoke about like difficulty with recruitment and how some people sort of, obviously some people died, but some came back and said, no, we don't want to be part of the study anymore. So I just wanted to find out in terms of your recruitment and consent, what type of consent do you use? Do you use broad consent? Can people come back and say, actually, even for the first phase, I want my data sort of erased, or you can't use my data anymore? So what type of consent do you use? And how are you going to sort of navigate that difficulty of retaining participants in your study? So that was for Michelle. And for Andrew, you use like different words such as African, Black, Asian, European and white and Black African. And in my mind, I was thinking about how I've read a few articles and saw a few documentaries about how there's a debate in the NIH about the usage of like race, ethnicity, and nationality, or particularly in genetic studies. So when you say African, what do you mean? Because I mean, I'm African, Michelle is also African. Is it geography? Is it race? Is race real? Is race not socially constructed? So when you say Black again, in South Africa, Black can be colored, Indian, or anything that's not white. So how do you use and how do you navigate sort of the debate around the issues of race and ethnicity, as well as identity, particularly in genetic studies? I'll stop there. Thank you.
Thanks, Pabala. You've given me the easier question, and Andrew can chew on the other one in the meantime. So just to say, we have used consent, which will allow us to, in the future, use both the samples and the data and use it for different projects. So all of this happens under ethical oversight. Research participants are always voluntary participants, and I say sometimes the fact that we do get refusals means that the process is working, because people should be comfortable enough and not feel that they're coerced to take part in the study. So I think that is a positive thing. You know, I think a lot is also about building up trust in a community. And it's about external factors sometimes beyond your control. Like, for instance, if there is a population which is overstudied and participants feel that they're being used for multiple studies, they kind of feel this is enough now, I've given enough of my blood. So I think we have to be very careful in terms of our community engagement and how we interact with our participants for the best possible outcome for both us and them. And, you know, in AWI-Gen, we now have a process of community engagement and feedback of results, where the engagement starts before, during, and happens afterwards as well. So we're hoping that through that mechanism, we will retain a lot of our cohort and we'll be able to do even a third wave of data collection in the future. Thank you.
Thank you, Michelle.
Yes, to try and address your question, thank you for your question. I think it's a very important one, but a very, very difficult topic to handle because I know it's very sensitive. And as you mentioned, I did use a lot of different terminology, and hopefully I didn't use any terminology that anyone would construe as offensive, because I think this is a hot topic and The literature, obviously in the genetic literature, because there is no biological definition for race, it is a social construct, not a biological one. So it does blur the lines a lot. So when it comes to working with ethnicity in our studies, we use the government category of Black African, and that is separated somewhat from other mixed ancestry categories. For example, in South Africa, we have a category called colored, which covers mixed ancestry individuals, but all of our participants self-identify their ethnicity. So it's entirely up to the participant how they self-identify. And obviously, when it comes down to the genetics, we use particular thresholds to cut out individuals who we think have divergent ancestry. But it is a very, very difficult topic, and I understand it's difficult to settle on a defined set of words and categories to work with something that's quite nebulous in terms of being a social construct like race. So I hope that sufficiently addresses your question, but I agree it's a very sensitive matter and it does need further debate.
Okay, thank you so much for that, Andrew, for your honest answer. And I think obviously maybe in your paper, if you can just, for like a few lines, discuss that, because I think it's very important. You can just obviously, again, how we use it in South Africa is different to the US as well, or globally. It's a debate and it's context specific. But I'm looking at the time as well, so I don't want to take too much time. There's about 2 minutes left. I see the comments are going, it's very hectic in the comments. There's people asking questions and saying, great presentation, and I have to agree with them. I'm going to read. So I see Olayinka, Jeff is answering your questions, so I'm not going to ask those. And I will follow up with you, Olayinka, about internships. I see a comment from Wrongling saying, since several IHCC members are willing to contribute data for COVID-19 studies, I'm wondering if any of you can lead a collaboration. So that's a question from Wrongling. And I don't know if that brought space or was that specific for Andrew and Michelle. Wrongling, was that specific to Michelle and Andrew or just anyone in the audience?
Yeah, Michelle and Andrew will be great. And the one in the audience can lead this one. That would be great. I think IHCC has potential since people wanting to contribute the data, not sample, but contribute the data for the study. I think that would be great.
Thanks, Wrongling. I think, maybe, the IHCC is a good place to disseminate information and ask for collaboration. So maybe what we do need to do is to put up our hands and say, please, if there's anybody who is interested in collaboration, the point was made earlier that we need replication data sets, and the Qatar genome study sounds amazing. So I think actually, saying we're looking for collaboration and actively seeking it is a good strategy to follow.
Thank you. Yeah, since you have already started, you and Andrew have already started. Yeah.
Okay, so in the interest of time, we can go on for another hour, but I do understand that people are busy. This was really, really powerful and interesting and engaging. I mean, you can tell we literally just had 30 minutes of question and answer and discussion as well. I just want to say to Andrew and Michelle, really great presentation, really great and important work that you are doing. We appreciate your time. We appreciate your efforts. To the audience, so much for joining us for our February webinar. We'll be advertising another webinar as the education team for March. So be on the lookout on our website and on social media as well. But I just want to say, enjoy your day, everybody. And thank you so much to Michelle and Andrew once more, because I really think this was one of the greatest webinars that we've hosted before. Thank you so much and good luck with the rest of your research.
Thank you very much. Goodbye.
Thank you very much.
Thank you for presenters and also for Barbara.
Thank you.
Bye.